from First MSc Biotechnology, KGC College. And the topic what I'm going to present or what I'm going to talk about is post-transcriptional modification. Let me give a small introduction about what is the information flow in biological systems. And the central theory or the central dogma refers to the flow of the genetic information in biological systems. And this is general and the genetic information where there is a flow from DNA to RNA and to a protein. This DNA, this encodes for genetic information for in most of the species, especially in eukaryotic species. It is most common, it is RNA in species like microorganisms and this bacteria and micro small prokaryotes, and it is most common for DNA in eukaryotic organisms. This is about the central dogma, the information flow in biological systems, and this is the diagrammatic representation of a DNA or a gene, which is transcribed or passed into a message and to a protein. This is what's happening in a biological system, and this is about the first step, which is the transcription. It is the process of synthesis of RNA from a DNA template. This is what is happening, and this transcription, whereas in prokaryotes or eukaryotes, it is in prokaryotes. It's not a complex process. In eukaryotes, it's a very complex process, and it has three main events, which is the initiation, the elongation, and the termination process. I'm going to talk about only the transcription or the translation because my post-transcriptional modification comes under the after the transcription occurs. So my speech is going to be a little more specified on the transcription only. And let me give an introduction about what is transcription. And DNA transcription occurs in a cell's nucleus. And this occurs in a eukaryotic cell, and this RNA that is synthesized from the DNA in this process is transferred to a cell's cytoplasm, where it is translated into a protein. This transcription process occurs in a nucleus, and this escapes or transfers from the nucleus to the cytoplasm for the further translation to occur. And in between this process, the post-transcription modification occurs, that what I'm going to talk about in the preceding slide and the coming up slide. In the transcription in prokaryotes, the RNA that is synthesized during DNA transcription is ready for the translation process to be translated into a protein. There is no other process to be required in prokaryotic, whereas in eukaryotic RNA from the DNA transcription is not immediately ready for the translation process to occur. So there is an intermediate post-transcriptional modification, the process which is required for the eukaryotic DNA or the RNA which is transcribed. There is an intermediate process to be taken place to make that mRNA a functional RNA, mRNA. This is about transcription, and I'm going to talk about the post-transcriptional modification of RNA, which accomplishes two main things. This modification helps the RNA molecule to be recognized by the molecule that mediate the RNA translation into proteins. During this transcription processing, portions of the RNA chain that are not supposed to be translated into proteins are cut out of the sequence. Let me give a small brief about what is post-transcriptional modification. This transcription and translation takes place in prokaryotes concurrently in bacterial cell. When the 3' end of an mRNA is undergoing transcription, the ribosomes near the 5' end to the sequence called as Shine-Dalgarno sequence, and it begins the translation process. Since the transcription and translations are coupled in the prokaryotic, before the protein synthesis, the bacterial mRNA has a little opportunity to be changed. In contrast, in eukaryotic cell, the transcription and translation are segregated both in time and space. In nucleus, this transcription takes place, while translation transcription takes place in the nucleus, whereas the translation takes place outside of the cell. And this separation offers a change to modify this post transcriptional modification offers a chance to modify the eukaryotic RNA before the translation process. These changes are made in the RNA molecule at the 5' end and the 3' end and to the protein coding portion. This is what is happening in the eukaryotic cell. Let me give a little more brief about why it is done. This post modification process is done. This post transcriptional processing helps to increase the efficiency of protein synthesis, allowing only specific protein coding RNA to go on to be translated. And without this process, this protein synthesis could not be significantly slowed since it takes much longer for a translational mechanism or machinery to recognize these RNA molecules because of this unwanted regions or non-coding regions in this uh, primary mRNA or the pre-mRNA, which is not functional. Significantly, more mRNA would have to be unnecessarily translated to achieve the same results. So this post-transcriptional modification is much required, and this is why this post-transcriptional modification is done. And these are the reasons. And I'm going to talk about what is the difference between an eukaryotic and a prokaryotic transcription. And in eukaryotes, this transcription and translation occurs in separate compartments. It has separate compartment. This transcription occurs inside the nucleus of the cell, and this translation occurs outside the nucleus or in the cytoplasm of the cell. Whereas in prokaryotes, this transcription and translation occurs simultaneously in the cytoplasm because it does not have a separate compartment. You know that it does not have a prokaryotic cell does not have an nuclear membrane. So this two process transcription and translation occurs only in the cytoplasm. And whereas in eukaryotes, the mRNA is polycistronic. That is one mRNA. This codes for more than one polypeptide. This is the term for polycistronic. Whereas in prokaryotes, this mRNA is usually monocistronic, which means one mRNA codes for only one polypeptide. In eukaryotes, the processing of mRNA is required in this eukaryotes for the maturation of the cell. Whereas in prokaryotes, no processing in eukaryotes is taking place because the mRNA matures on the transcription itself. So, so a post-transcriptional modification is not required in prokaryotic cell. This is the primary most difference of eukaryotic versus prokaryotic transcription. And let me, this is the schematic representation of the difference between a prokaryotic and a eukaryotic cell, where we see here the prokaryotic transcription and translation are coupled each other, whereas in eukaryotes, this mRNA processing and the transport of out of the nucleus, this where we see here, this DNA is unwinded or processed or transcribed into a protein and comes out of the cell and becomes and come out of the cell into the cytoplasm for the translation or the protein synthesis translation to occur. This is the primary most difference. And let me move on. What happens from a DNA? Transcription takes place, and a primary mRNA or the pre-mRNA, which is not functional. This is what I'm talking about in eukaryotic cell. There is a primary mRNA or pre-mRNA, which is the primary transcript also. There are three processing taking place, which indeed process into mature RNAs, which are the RNAs tRNA, the transfer RNA, and mRNA, which is the messenger RNA, and the rRNA, which is the ribosomal RNA. And let me talk about it individually. And let me talk about what is mRNA, which is the messenger RNA. This messenger RNA operates as a template for the protein synthesis to take place. And this mRNA, which is the messenger RNA, encodes genetic information from a DNA as a transcript and translates the information to a ribosome and helps to assemble the amino acid in a proper and a sequential order. There are three main regions occurring in both prokaryotic and eukaryotic mRNA, which is the 5' UTR site and the protein coding region and the 3' UTR site. These are the three regions where this mRNA operation or the template for protein synthesis take place. And let me move on the mRNA processing. What happens here? The primary mRNA or the pre-mRNA or the primary transcript processed first and then are exported from the nucleus of the cell to the cytoplasm. The mRNA fate is to be completed or regulated degradation. There are three main steps undergone in the mRNA processing, which is the post-transcriptional modification. The first step is the 5' capping, the splicing, the poly-A tailing, or the poly-A cap addition, which I'm going to talk about individually in a elaborated manner. Let me move on to the 5' capping. And here, this is one type of eukaryotic pre-mRNA modification, or in which there is an addition of a structure called as 5 cap at its end. At the 5 cap end of mRNA, the cap consists of an additional nucleotide and a methyl group called CH3 at the base of one or more nucleotide. At the 5' end of a newly inserted nucleotide and the 2-OH group of sugar. This is where it attaches. This occurs after transcription initiation, and this insertion of cap takes place in a very quicker manner. It is possible to represent the 5' end of the pre-mRNA as the 5-PPP NPN, where in which a ribonucleotide is represented in the ribonucleotide is represented as the capital N, and the phosphate, which is represented as small p. And one of these phosphate group is removed shortly after the start of transcription, and a guanine nucleotide is added subsequently, and a special 5' bond connects this guanine nucleotide to the pre-mRNA, which is somewhat quite different from the normal 5'3' phosphodiester bond that binds all other RNA nucleotide here. To the 5' end, one more methyl group is added. This is another process, and the first of these methyl group is added to the 7th position of the base of the terminal guanine nucleotide, making the base as where you see here the 7-methyl guanine. 7-methyl guanine. Next, in the 2nd and 3rd nucleotide, a methyl group may be attached to the 2' position of the 2' position of the sugar. And additionally, this methyl group can rarely be attached to the bases of the 2nd and 3rd nucleotide of the pre-mRNA. This is what is happening in 5' capping. This is the steps involved that I was talking about, where we see all this methyl transferase will add a methyl group at the 7th position of the guanine base. This is the primary most steps happening. And let's move on to the functions. What this 5' capping, what are the functions of this 5' capping? This protects the mRNA or the pre-mRNA or the primary transcript from the ribonuclease degradation, and it stabilizes the mRNA, the messenger RNA, and this enhances the translation and the splicing process. This is the second process which is occurring for the post-transcription modification, and there is an enhanced transport from the nucleus to cytoplasm due to this 5' capping. These are the main functions of 5' capping. Let me move on to the second process called as splicing. The removal of introns, that is splicing. The removal of introns by RNA splicing is another major type of eukaryotic pre-mRNA modification. Here, before the RNA moves to the cytoplasm from the nucleus, this modification takes place in the nucleus itself. And the presence of 3 sequences in the intron is required for splicing. Here, what takes place? One end of the intron is transferred to the 5' splice site, and the other is the 3' splice site, where the short consensus sequence they exist on these splicing sites, where most introns begin with GU in pre-mRNA and the end of AG, which is the adenine guanine. And the third sequence that is necessary for splicing is present at the branch point, which is an adenine nucleotide that is situated in the 18th to 40th nucleotide upstream to the 3' end, where we see here in the 3' end splice site. This splicing occurs within a structure called a spliceosome, which is one of the most and most complex of all molecular machineries or complexes. And these are the functions of snRNP, recognizing the 5' splice site and the branch site, bringing these sites together and catalyzing or helping to catalyze the RNA cleavage. And let me explain about the structure of what is spliceosome complex, which is the snRNP. It has six regions, which is the U1, which is used for the 5' site recognition, and the U2 site where the branch for the branch site recognition, and the U4, which is formed for base paired complex and acts or reacts with the U6 complex, and the U5 complex, which the 3' binding of U4 and the U6 complex. This helps for this, and the U6 complex with this makes complex with U4, which makes the spliceosome transesterase, which is an enzyme. And these spliceosomes recognize the intron starting with 5' GU and ending in AG, adenine guanine, at the 3' site. I repeat, the 5' at GU at the 5' site and AG in the 3' site. This is what the spliceosome complex, and the process that RNA splicing occurs, that an intron is between an upstream exon 1 and downstream exon 2 before the splicing takes place. And in these two distinct stages, the pre-mRNA or the primary transcript is spliced. The pre-mRNA is cut at the 5' splice in the first stage of splicing. This cut frees, and where we see here the exon 1 from the intron, and the intron's 5' end connects to the branch point, and this intron folds back on itself, creating a structure called as lariat. This spliceosome is a very key in cutting this reaction to take place. In this reaction, via an transesterification reaction, the guanine nucleotide in the consensus sequence at the 5' splice site binds with the adenine nucleotide at the branch site. And we can see the branch site here. This is the branch site, which is common in this site to the cytoplasm, where there is a translation occurring. The mature mRNA consists of the exons spliced together is exported. A cut is made at the 3' site, 3' splice site in the second step of mRNA, and simultaneously at the 3' end of exon 1, which is covalently connected or spliced to the 5' end of exon 2. And it releases where you see here, it releases the intron as a lariat, with the region what I was talking about. And when the bond splits at the branch site, where we see here, it splits to make a mature mRNA. The intron becomes linear, and then it is quickly degraded by the nuclear enzymes, and to the cytoplasm, where it is translated. The immature mRNA consists of only where we see here, the unwanted introns are cut away with the spliceosome complex, and only the exon which are spliced together is exported from the nucleus into the cytoplasm for the further translation process to undergo. This is about the RNA splicing, and let me talk about the alternative splicing. And in order to generate alternative forms of mRNA, a single pre-mRNA is processed in various ways, resulting in the development of various proteins from the same DNA sequence. And an alternative splicing in which the same pre-mRNA, the primary transcript, can be spliced in more than one way. Where you see here, it is spliced in more than one way into protein A and protein B to generate multiple mRNAs that are translated into different amino acids or different proteins. And thus different proteins, if different amino acids are synthesized, thus different proteins are synthesized, and thus it is an alternative form of processing the mRNA. And another method of alternative splicing process involves the use of several 3' cleavage sites where the pre-mRNA comprises two or more potential cleavage and polyadenylation adenylation sites in the same pre-mRNA transcript. Both alternative splicing and multiple 3' cleavage sites can even exist. In multicellular eukaryotes, especially in V eukaryotic cell, alternative splicing in pre-mRNA processing is very much common. And recently, many researchers predict that more than 90% of our human genes undergo this alternative splicing, and this splicing varies between human. It also varies between human tissues as compared to other cells. Only the human brain and liver tissues have more alternatively spliced RNA through this alternative splicing process. And let me move on to the third process, which is the polyadenylation or the poly-A addition. And the addition of 50 to 250 or even more adenine nucleotides at the 3' end, forming a poly-A tail, is a second kind of modification, and which is a kind of modification to the this is the third kind of modification to eukaryotic mRNA. These nucleotides are not encoded in the DNA, but are inserted in a process called as polyadenylation, which is followed by the transcription process. And the enzyme which is involved here is RNA polymerase 2, which is transcribed in eukaryotic genes are transcribed well past the end of the coding sequence. Much of the extra, which is cleaved at the 3' end and the poly-A tail, is inserted back. And this is the steps which is involved. The sequences both upstream and downstream of the cleavage site are necessary for the processing of 3' end of the pre-mRNA. Generally, the downstream of the cleavage site is a sequence rich in uracil nucleotides, or even in guanine and uracil nucleotides. On many mRNAs, the poly-A tailing, the mainmost function, or it increases the stability and increases the time duration in which the mRNA always remains intact and available for translation for various other enzymes to. It does not allow various other enzymes to degrade it, and this poly-A tail also enhances the ribosome's attachment to the mRNA. And these are the main steps which are involved in polyadenylation: the cleavage of mRNA at the 3' end and addition of adenine residues. And these are the specific sequences. A cleavage sequence has a CA, which is the cytosine and adenine. This polyadenylation sequence are AA, adenine, adenine, uracil, AAA, and adenine, adenine, adenine. A represents adenine and U represents uracil, which are located at the 10 to 30 nucleotide upstream to the cleavage site, and which is highly conserved. And this GU is this GU-rich sequence present at the 20 to 40 nucleotides downstream to the cleavage site. This is the specific sequence in poly-A tailing. And after these three processes, the splicing, polyadenylation, and these processes occur the post-transcriptional modification. There is an matured RNA, mRNA, which is synthesized, where you see here, this is the without any unwanted sequence or unwanted introns in it, and the unwanted non-coding regions. This is mature mRNA, which consists of the 5' cap, where we see here, and the 5' untranslated region, or the 5' UTR, and a start codon with the coding sequence, which is the CDS, the stop codon, the 3' untranslated region, and a poly-A tail at the 18th site. This is an mature mRNA. This is all about mRNA processing. Let me move on to the transfer RNA, which is the tRNA. This is also called as a soluble or supernatant or adapter RNA. And this tRNA is smallest among RNAs, which is of only 75 to 93 nucleotides. This recognizes codon on the mRNA. This structure shows high affinity. This is the structure of tRNA, which shows high affinity to the amino acids, which carries amino acid to the site of protein synthesis. This by the name, the transfer RNA. we can know that it carries something or transfers something. It carries amino acid to the site of the protein synthesis. This tRNA is transcribed by the protein by the enzyme called as RNA polymerase 3. I repeat, the RNA polymerase 3. The tRNA genes also occur in repeated copies throughout the genome and may contain the introns also, which are the untranslated or the non-required regions or the non-coding regions. And let me talk about the processing of tRNA, which is the um, removal of leader sequence and the tailor sequence. And replacement of nucleotides. This modification of certain bases undergoes three processes, which is the replacement, which in this there is a replacement of uracil residues at the 3' end of pre-T transfer RNA with SCCA, cytosine adenine sequence, and addition of methyl and isopentenyl groups to the heterocycle ring of the purine base. And the third process, which is methylation, where there is a methylation of 2-OH group in the ribose of any residue, and there is a conversion of specific uridines. U represents uridines, not uracils. The uridines to dihydrouridine and pseudo-uridine, which is uridine-like, which is a pseudo-uridine, which is not a true uridine. And there is an existence of an intron 20 at the 20th base. And this is what I was talking about, the tRNA processing, where there is a primary transcript and intermediate, and to a matured tRNA, which undergoes a process called as replacement, addition, and methylation. This is about the processing of tRNA. And let me move on to the ribosomal RNA. And in a cell, 80% of the there is more than 80% of rRNA, which is the ribosomal RNA, which serves to release the mRNA from the DNA. This acts as ribozymes in the protein synthesis. Ribozymes. It is relatively very rich in the guanine and cytosine content. Is very rich. Where you see here this the schematic representation of difference between a prokaryotic ribosome and a eukaryotic ribosome. This prokaryotic ribosome consists of a 70S structure, where it is divided into two structures called as 50S. This 50S is then even more divided into 23S and 58S, which consists of 31 proteins. And this 30S subunit, it consists of 16S, which consists of 21 proteins. And this is the eukaryotic ribosome, which is an 80S structure, which is divided into a 60S and a 40S subunit. This 60S subunit consists of many parts like 28S, 5.8S, and a 5S, which consists of 50 proteins. And the 40S, which consists of 18S and consists of a 13 protein. This 18S is a subdivision of ribosomal eukaryotic 40S subunit of the ribosomal RNA. And let me move on to the processing of ribosomal RNA. This processing of 40-base RNA molecule occurs only inside a nucleus. This structure is inside the nucleus, where we know that inside the nucleus there is a structure called as nucleolus. This rRNA, the processing takes place only in the nucleolus. And the 45S molecules they are tightly associated with the protein forming, which is the RNPs. And there is three processes which are occurring. The first cleavage, first cleavage. This occurs at the site 1 and removes the 5' terminal sequence and provides intermediate and an 18S. And there is a second cleavage, which occurs at the 41S intermediate at the site of 3', which generates the 32S intermediate. And the final cleavage, where there is a separation of 32S intermediate into 28S and 5.8S, which is further processed with the RNA into a 28S, 5.8S, and an 18S. This is the processing of ribosomal RNA, what I was talking about. And the lastmost topic is the synthesis of 5S rRNA, where the DNA cistron for the 5S rRNA is present outside the nucleolar organizer. This transcription requires three enzymes called as RNA polymerase 3, which you all know about, and the TF3A, TF3B, and the TF3 transcription factor C. TF is the transcription factor. These are the enzymes or the factors required for this process to take place, and this is the end of the post-transcriptional modification and the processing of the mRNA, tRNA, and rRNA. What I was talking about for this long. And let me move on to the conclusion of what my specific topic, which is the post-transcriptional modification, which is a set of biological process common to most eukaryotic cells. What is talking, prokaryotic cell does not require a post-transcription modification, because transcription and translation is a coupled process, and it is not a complex process. Only eukaryotic cell requires this, and this is most common in eukaryotic cell, by which an RNA primary transcript or the pre-mRNA is chemically altered following from the transcription which is occurring in the nucleus, producing a mature, or from an non-functional mRNA to a functional RNA molecule that can leave the nucleus and function for and perform any variety of functions inside the cell, particularly from the transcription process occurring inside the nucleus, outside, transported outside the cell for further translation process to occur. This is the end of my seminar, and these are my references. Thank you.